First, I would like to thank the coordinating committee to give me this opportunity to present our work at UBI Asia entitled UB-612 pan-SARS-CoV-2 T-cell immunity promoting vaccine protects against COVID-19 moderate to severe disease. The COVID-19 pandemic from the beginning of 2020 until the end of 2023 has officially claimed 7 million lives globally. But the actual death toll from the disease may be triple, close to 21 million, said recently by the WHO. This tragic catastrophe has also brought in countless vaccine injuries, long-haul COVID, long vaccine syndrome, and excess deaths. If we as human beings had abided by ethics and morals, the greatest catastrophe in this century could have been avoided. This presentation will address two key points. First, why and how a pan-SARS-CoV-2 vaccine should be prepared. And second, why and how our UB-612 vaccine can clinically protect against infection and moderate to severe disease, which in other words, hospitalization and ICU admission. The SARS pandemic may be far from over for two reasons. Firstly, gain-of-function scientists continue to create hyannis viruses as evidenced by publications. For instance, the precursor of the SARS-CoV-2 virus leading to the COVID-19 pandemic catastrophe, the new Boston chimeric SARS, killing over 80% of mice, and the COVID-like pangolin virus, causing 100% mouse mortality, are being created. The purpose behind these man-made actions is now fully understood, whether for the good of mankind, vaccine commercial interests, or biologic warfare. Secondly, using suboptimal mRNA vaccine platforms and without targeting non-spike proteins, these vaccines produce suboptimal immunity with safety concerns, which, upon reflection, frankly, should not have been used for massive vaccination across all age groups for public health purposes. The immune-evasive XBB sublineage, for example, HV1, EG5, has given way to BA 2.86 sublineage, from which the offspring JN1 variant is now dominating. Hospital admission cases have surged since WHO declared the end of the health emergency on May 5th last year. Undue boosting with suboptimal vaccines can foster viral mutations, which can occur on both the spike and non-spike envelope membrane nucleocapsid proteins. These mutations could also escape surveillance, not only by B cells, but also T cells. It is important to note that JN1 variant has the highest number of mutation sites to date, with one more mutation site at the S protein amino acid sequence 455. Could JN1 or subsequent mutations make the virus more fusogenic, thus opening the door again to allow formidable systemic infection? Despite virus mutations, identification of highly conserved T-cell epitope sites is crucial for designing an ideal pan or universal SARS vaccine. Our UB-612 vaccine, constructed in the first quarter of 2020 when the COVID outbreak began, utilizes 5 conserved T-help CTL peptides, 3 on the spike 2 protein and 1 each for the membrane and nucleocapsid proteins. When encounter virus infection or are stimulated with vaccine booster, the ensuing immune response in a vaccinated host could promote a full-blown and broadly recognizing helper and cytotoxic T-cell immunity. Thus, UB-612 offers the vaccinee a precise and effective response against all SARS variants, including the pangolin virus version. The only exception among all conserved sites with small differences is the site marked in red, as shown on the slide. Along with these 5 specific T epitope peptides, and together with one highly promiscuous and proprietary UB peptide to serve as a catalyst immunogen, UB-612 promotes a full-blown T-cell immunity. For production of B-cell immune antibodies, UB-612 is equipped with a monomeric S1 RBD subunit protein with Wuhan, the original ancestral strain, as an immunogen, which is linked to single-chain Fc of immunoglobulin molecule to support its stability. UB-612 contains a trace amount of CpG as a stabilizer to bring together the 6 positively charged T-cell peptides through charge neutralization, which are formulated with adjuvants as the adjuvant. The complete UB-612 vaccine product resembles, in this cartoon, a ball-like virus. UB-612 is unique as it simultaneously targets epitopes on both spike and non-spike proteins, producing synergistic, potent B-cell and T-cell immunity. Now, let's examine the UB-612 vaccine's real-world performance in a clinical setting, focusing on its protective effect against any infection and moderate to severe disease, specifically hospital and ICU admissions, and the biochemical mechanism behind the protection. In the years of 2020 and 2021, Taiwan effectively managed to keep SARS-CoV-2 virus at bay, reporting extremely low infection cases with an exception of a brief surge in Alpha variant cases confirmed between April and July 2021. However, a year later, in late March 2022, a sudden outbreak of the highly contagious Omicron BA.2 and BA.5 variants occurred, persisting through the end of the year. A large-scale phase 2 trial in 2021, approved by Taiwan FDA, provided valuable opportunity for UB-612 to assess its protective effect during this precious time window. On May 11, 2022, a 6-week window from late March 2022, which equates to 6 to 8 months post-booster, was employed during this Omicron outbreak for the assessment of protection from any infection. On September 30th, a 6-month window from late March 2022, which equates to 10 to 12 months post-booster or 14 to 18 months post-second dose, was employed for the assessment of protection against hospital and ICU admission cases. Out of the 2,900 enrollees who completed the questionnaire survey, only a limited number of participants adhered to the UB-612 vaccination regimen. That is, only 103 participants received the primary 2-dose series, and an additional 212 participants receiving both the primary 2-dose series and the third UB-612 homologous booster dose were assessed. This limitation was primarily attributed to the official Taiwan CDC recommendation to the Taiwan public to receive only EUA-approved heterologous booster over time. As of May 11, 2022, 6 weeks after major Omicron outbreak, Participants receiving 3 doses of UB-612 only reported a 1.2% infection rate, in contrast to a 2.2% rate in the Taiwan population receiving EUA vaccines. As of September 2022, 6 months after the Omicron outbreak, both participants receiving 3 doses of UB-612 and overall Taiwan population receiving EUA vaccines reported an approximately same infection rate of 28%. As of September 30th, 2022, all 103 participants receiving only the primary 2-dose series and over 14 months after the dose reported none, means 0%, of hospital and ICU cases. Similarly, among the 212 participants receiving 3 doses of UB-612 and observed for at least 10 months post-booster, 0, means none, reported hospital and ICU cases. In contrast, those who already on 2 or 3 doses of UB-612 but took 1 or more EUA vaccine doses as booster, few are observed to report moderate to severe disease. This implies that if you insist on only receiving the UB-612 vaccine, you will probably not get moderate to severe disease. As of September 30th, 2022, notably, 65.4% of the Taiwan population has received 3 doses or more of EUA authorized vaccines, which are mainly from Moderna, Pfizer, and AstraZeneca. In this population, there were hospital and ICU admission rates reported in a dose-dependent manner, ranging from 0.44%, 0.22%, 0.11%, to 0.06% for individuals unvaccinated receiving 1, 2, or more or equal than 3 EUA vaccine doses, respectively. In contrast with UB-612's 0% cases of hospital and ICU admission, we thought the differential performance in disease control could be explained in the vaccine design. UB-612 has multiple immunogens, using multiple targets on both spike, namely monomeric S1 RBD subunit protein, and 3 sequence conserved epitope peptide on S2, and on non-spike, namely sequence conserved epitope peptides from membrane and nucleocapsid proteins. All other vaccine platforms using the trimeric full-length spike protein as the sole immunogen, which could produce many non-neutralizing antibodies, could be potentially harmful to immunity and safety. The vaccines with spike only as a sole immunogen will produce limited T-cell immunity. We anticipated a tendency that UB-612 would generate higher B-cell neutralizing antibody level and stronger, durable T-cell immune responses. In the clinical phase 1 study after the 2-dose primary series, we observed only a mediocre anti-wild-type live virus neutralizing titer around 108. Yet this level declined with an unusually long half-life of 187 days. After the booster, 3rd dose, the anti-wild-type neutralizing titer rose sharply by 37-folds with a titer of nearly 4,000. UB-612 induces high cross-neutralizing antibody titer against the Delta virus with a neutralizing titer of nearly 2,400, which tops the whole vaccine list. This implicates that, in regard of neutralizing antibody titer strength, UB-612 has a competitive edge over other vaccine platforms. a model bridging anti-RBD IgG to vaccine efficacy against COVID-19 symptomatic infection caused by the Wuhan wild-type strain reveals that UB-612's protection efficacy after 2 doses and 3 doses, without regarding the benefit of T-cell immunity, to be 72% and 95% respectively. If the bridging method is applied for vaccine efficacy to combat Delta strain, it is legitimate to assume that UB-612's performance will be advantageous in protection efficacy over other vaccine platforms. In a clinical phase 2 study, UB-612 booster shows substantial live virus neutralizing antibody titers against Omicron BA1, BA2, and BA5, all of which surpass the counterparts in other vaccine platforms. We are curious why, apart from the BA1 variant, no other EUA vaccines have provided other virus neutralization data. It is worth noting that the neutralizing titer of the EUA vaccine against the BA1 virus is generally below the low value of 100, which is close to the detection limit. Testing the same serum sample set against BA1, BA2, and BA5 using the pseudovirus assay, UB-612's neutralizing titer strengths also showcased competitive edge over Novavax, Pfizer, and Moderna vaccines. The neutralizing titers from 4 other vaccine platforms are even far lower. Up to this point, UB-612 booster vaccination has shown a potential competitive edge over other vaccine platforms in terms of serum viral neutralizing titer, either by live virus or by pseudovirus assay. Therefore, after UB-612 vaccination, there exists a good correlation between live virus and pseudovirus neutralization assays against BA1, BA2, BA5. Beyond BA5, a 10-fold decline in neutralizing titer for XBB 1.5 was observed, indicating the virus increasing immune evasion. A recent study has suggested that the neutralizing antibody titer against the new JN1 variant is around 2 to 5-fold lower than XBB 1.5. In addition to producing high viral neutralizing antibodies, the T-cell response after UB-612 booster vaccination displayed even more competitive edges in contrast with other vaccine platforms. By interferon ELISpot analysis, the T-cell response after 2 vaccine doses is highly potent, measured at 374, which is quite durable as it retained 70% at 6 to 8 months post-second dose, namely pre-booster, measured at day 261. After booster, it increases to 444. By estimation, the conserved epitope peptide pool without RBD contributes about 75% of the overall memory T-cell immunity. Alongside these findings, there was a notable presence of approximately 2 to 4% cytotoxic CD107A plus, granzyme B plus, CD8 positive T cells, stimulated by the antigen pool of T helper CTL peptide plus RBD. We're now addressing memory CD8 positive T cells that are crucial in the control of disease severity. We investigated with 7 available study participants. 4 infected assigned 1 and 3 non-infected assigned 0. UB-612 vaccination demonstrated that by Spearman coefficient, infection severity correlates well with memory CD8 positive T cells, interferon gamma positive or CD107A positive, granzyme B positive, but not with CD4 positive T cells, interferon gamma positive. Notably, no relationship between disease severity and viral neutralizing antibody titers over time could be established. While UB-612 vaccination induced neutralizing antibody potency has shown potential competitive edges over all EUA vaccines, how about T-cell responses? Regarding memory T-cell responses, the competitive edge are even more pronounced. In a 3-dose homologous boosting, clearly the other vaccine platforms produce low levels of interferon-producing T-cells, and it gets worse when a second booster, which is 4th dose, is administered. This implicates that the more the doses, the lower the T-cell immunity that can be recalled. Could this mean a T-cell exhaustion, which would allow a higher chance to get reinfection? The answer may be yes. Data on this slide is demonstrated by a BA.5 bivalent mRNA vaccine given to individuals who received 1 or more monovalent vaccine doses. These individuals were uninfected or previously infected. The more monovalent vaccine doses they previously received, the higher risk they are to be infected or reinfected after bivalent BA.5 vaccination. These results are consistent for the BQ and XBB variants that were dominating at the consecutive time frames. Note also, the protection effectiveness levels are low at 29% against BA.5 and even lower against BQ and against XBB variants at 20% and 4% effectiveness respectively. Altogether, the more prior mRNA vaccine doses, the sooner the protection would wane. This is also evidenced in the case of a hybrid immunity clinical trial described next. For COVID infection, we by now know that hybrid or natural infection versus EUA vaccine immunity protects better than natural infection immunity. If you were infected before, a follow-up 2-dose primary vaccination will provide protection against hospital admission or severe disease for at least 12 months. However, we found that a 3rd dose with 1 booster of mRNA vaccination presents less protection, which starts to decline at 6 months post-booster. One of the reasons could be that the previous infection provides immunity against spike and non-spike, yet the follow-up vaccination provides immunity against spike only, which does not match well with the natural infection immunity. It is important to know that there is an intrinsic gap in immunity constituency between natural infection and the currently used EUA spike-only vaccines. That means Pfizer, Moderna, Novavax, etc. Let's come back to vaccine-naive infection immunity. We by now know that natural infection immunity protects better than current spike-only mRNA vaccine-induced immunity. The reason is simple, because the immunity from infection targets both virus spike and non-spike proteins, while the mRNA vaccine targets spike protein only. Note that the immunity from previous non-Omicron infection can provide 90% effectiveness against severe disease caused by Omicron BA.1 infection, at least for 40 weeks, and the protection wanes very slowly. Clearly, as compared to vaccine immunity, the natural infection immunity protects much better against Omicron infection, be it against severe or mild symptomatic infection. Potential deficiencies in protecting immunity and effectiveness observed with current spike-only mRNA vaccines can be corrected by utilizing a vaccine like UB-612 that targets both spike and non-spike proteins. As shown in this slide, in a phase 3 trial, UB-612 has proved to be a competent heterologous booster for currently EUA vaccines, enhancing in a defined anti-BA5 seroconversion rate for Pfizer and AstraZeneca by 12% and 13% respectively. The enhancing effect is even 60% better for the virus inactivated vaccine BIBP. Furthermore, in the same phase 3 trial, UB-612 as a heterologous booster can enhance viral neutralizing effects against BA5 by 1 to 6 folds for currently EUA authorized vaccine platforms. Overall, the pan-SARS-CoV-2 UB-612 protein peptide multitope vaccine may serve as an effective primer and booster for persons at risk of COVID infection. There remains an outstanding concern over the vaccine efficacy of currently authorized mRNA-based vaccine platform, in particular regarding a high production of IgG4 upon repeated administration. There is a delayed but drastic process after the boosting third dose. It has been found that IgG4 production remains high over the fourth dose that ends up with an increasing ratio of IgG4 over IgG1. The class switching to IgG4 antibody is known to be associated with a reduced capacity of the spike-specific antibodies to mediate antibody-dependent cellular phagocytosis and complement deposition, which, to the least, will be detrimental to the combat against virus. The sustained high IgG4 antibody level could be responsible for the cause of chronic immune evasion that necessitates frequent booster administration. There are serious adverse effects associated with mRNA-based vaccines, as shown in the left panel. This may relate to a pro-inflammatory action of lipid nanoparticles that carry the mRNA payload to penetrate into cells, tissues, blood-brain barriers, etc., where trimeric full-length spike protein is expressed and released, along with S1 fragment that circulate in the blood. Of most concerning will be the cardiovascular toxicity, which is shown in the right panel. It has been observed that vaccination is associated with an increase in myocardial FDG uptake, indicating a higher chance of heart injury as compared to the unvaccinated individuals. UB-612 is safe, well tolerated, and without any concerns of severe adverse events. UB-612 is the safest amongst all listed vaccines in each of all categories of adverse events, ranging from fatigue to headache. UB-612's adverse events are mostly of mild degree. Moderate events are seen in a small population, while all other vaccines are associated with substantial moderate and severe adverse events. Factors desired of a COVID-19 vaccine. It has to be a safe, well-tolerated, and without concerns of severe adverse events. It has to be a vaccine with immunity to mimic natural infection immunity. It has to be a vaccine that can safely build herd immunity with decent B and T cell immunity. It has to be a vaccine that can target effective conserved epitopes to mitigate mutation impact. The vaccine also should not count on a trimeric full-length spike protein as an immunogen, due to the concerns of non-neutralizing or enhancing antibodies along with other safety issues associated with the spike ACE receptor binding issue. The vaccine should allow T-cell immunity to act as the leading actor, not a supporting cast. It should be a vaccine that can serve as a substitute for the current spike-only vaccine. Summary on UB-612 COVID vaccine. UB-612 is a safe vaccine with immunity mimicking natural infection immunity. It targets S1-RBD and conserved T-cell epitopes on both the spike and non-spike proteins, producing neutralizing antibody titers and T-cell immunity with competitive edges over currently authorized EUA vaccines. It has 0 hospital and ICU admissions over 12 months and only 1.2% of any infection at 6 weeks after the Omicron outbreak. It offers potent memory cytotoxic CD8 positive T-cell immunity, which underpins the control of disease severity. UB-612 can serve as an effective primer and booster for those at risk of SARS-CoV-2 infection. Thank you for your attention.